Okay, welcome to the tools and techniques of immunology that we are using. So, in last class we are discussing about immunodiffusion. So, we purify antigen, we inject antigen to animal, we have some antibody, we hope some antibody is there initially, and then we check the presence of antibody in serum with a very simple technique. You just need Petri plate, agarose and your antigen and antibody to see the precipitin line, and we also discuss about why precipitin line is found, what is the logic of this precipitin line, why how diffusion work. But that can tell you that okay, the antibody production is going on, but it cannot tell you exactly how much amount or quantitation is there, right. So, if you want to quantitate any antigen or antibody, what we do is normally. So, normally what we are going to do this, we are going to use a very popular technique, many of you might have heard about that. In short, it is called ELISA. Okay. In short, it is called ELISA, ELISA. ELISA stands for what? That is also many of you know, but still I am writing. It is enzyme linked immunosorbent. I just wrote purposefully this S bigger, immunosorbent assay. Okay. Enzyme linked immunosorbent assay. That technique, I mean this technique is very popular and routinely used for many labs, in the pathological lab, for detection of many things like hormone detection, quantification. So, these particular techniques, I am just not going to go the experimental detail because it will take a lot of time and you may not need at this stage, but just going to tell you how you can do that. What you can do with this experiment? We can do first detection. Okay. What we detect? We can do detection of what? Detection of either antigen or antibody. We can detect both. Again I am telling you, if you need to do anything for antibody, you need to have antigen, and if you want to know something about antigen, you have to have antibody against that antigen. Okay. So, either one you can do, and second, mostly very importantly, what you can do is you can do estimate. Okay. You can estimation you can do. So, estimation of either antigen or antibody you can do. What is the principle? All the principle, general principle of all immunotechniques are antigen binds to antibody or antibody binds to antigen. They interact. So, their interaction we are going to detect. So, what we do here? What we do in ELISA? ELISA we do is actually we take a well. Okay, that is a plastic well. Normally you can do it in tube also, but normally we use 96 well plate. Okay, 96 well plate. We also call it micro titer plate. Micro titer plate. Okay. So, micro titer plate also, or either one, this is same thing. What it is there? It is there are 12 well, 1, 2, 3 up to then 12, and there are 8 rows. Okay. So, in one row there are 12, and at the top of the plastic it is written 1, 2 and then gradually 12, 1, 2, and this side in the column wise it is written A, B, C. So, then you have 8. So, total is 8 times 12. So, it is a 12 into 8 is equal to 96 well plate. Okay. So, 12 in each row, and there are 8 in 12 in each row, and there are 8 rows. Okay. In one row there are 12 wells, and if I see this well, big way. Okay. If I am just erasing this part. So, if I see one of the well like this big, what I will see is this well, this you have to buy, you have to call, like there are many 96 well plate for different purpose. Okay. So, you have to go, when you are buying, you have to plate ELISA plate. What is the difference between other plate? Because you can grow cell also in this 96 well plate. This is a format, 96 well is a format, but you have to buy 96 well ELISA plate. What is there? This is, in the case of ELISA, this well bottom or the wall of the well is coated with polyvinyl chloride. What is the purpose of this polyvinyl chloride? Because it can adsorb protein, because you know antigen is mostly protein that we are working with, and antibody is definitely a protein. Okay. So, if antibody and antigen is a protein, and PVC, what the role of PVC? Because antigen antibody can attach to it or adsorb with it. So, what we do? So, this plate is, or the well is PVC coated. Now, if I want to see that in a given solution if antibody is there or not, what I will do is, first I will add some pure antigen here. So, I will take antigen solution, okay, antigen solution I will add. Okay. So, if I fill this antigen, if I fill this with the antigen solution and keep it for some time, what will happen? Suppose the antigen color is red, okay, antigen is say like this, or make it circle, my life will be easy. So, then if you keep this antigen solution everywhere, it is everywhere antigen is fill up. Okay. So, there are a lot of antigen molecule here, and if you keep it, after certain time what will happen? After certain time means maybe 1 hour, maybe overnight, depending on how much antigen you have, what we will see is, we will see that antigen is attached with the bottom of the well and the wall of the well. Okay. So, antigen is attached with the wall, you cannot see anything, you just have to believe that antigen is attached. Then every step I am not going to repeat, because I am not going to discuss about the total techniques like how exactly it work, what is the problem, I will just tell you the principle. So, every time you use or we use any reagent, next step is you have to wash out, okay, because this solution, I mean the solution you added was full, right, whole well was full of that. So, what will happen? Some will attach here, okay, some will attach here, but lot of solution will be there. So, you have to wash with just phosphate buffered saline. Normally, we use PBS, okay, phosphate buffered saline. You can type this PBS and see what is the composition, is sodium chloride phosphate, because it is a saline after all. So, what is the composition? So, it is not important here. So, we wash with PBS. So, what will happen? All unbound and extra thing will be washed out. So, now when washing is done, when washing is complete, this is the case. Now, in this stage there are few things, we call it blocking, I am not discussing here, just see what is happening. In this stage, if suppose the antibody is let me change the color green. So, now I will add, in after this washing is done, I will add antibody solution, okay, that may be directly serum or diluted. Okay. So, now if that serum has the antibody against your antigen, what will happen? They will bind here, wherever antigen is there, they will bind. Okay. It depend. Now, I am just making it saturated. So, if antibody amount is well enough, what will happen? That will bind all the antigen available. If you have less antigen, I mean less amount of antibody, maybe some antigen will be free, it is not going to bound. Okay. So, now in this case I assume that all antigen is covered by antibody molecule. Even after that you incubate for some time, wash out. So, this is now the condition, you cannot see that, clear, because it is colorless. Now, what happened? Two possibility. Now, we have to detect the antibody, whether antibody is there or not, okay, because basically we are going to see the just a plastic. So, to do that, whether antibody is really attached to it, first thing is the antibody solution I added, it may or may not have antibody, because that is what we are going to test. Second, if it is there, how much it is there, both we can. So, then we have to detect. So, detection I am telling you first the simpler way, though it technically it is little more difficult. Suppose this antibody solution, that antibody, what I can do is I can make some attachment with it. Okay. What is this attachment? So, I make the antibody in such a way, or purify the antibody and modify the antibody such a way, I attach one enzyme. I attached one enzyme with this antibody. Okay. I attached one enzyme with this antibody. Clear? So, this antibody, this enzyme, again colorless, you can't see. So, when I am adding this, so every antibody has this enzyme. Okay. Every antibody was attached with this enzyme. That is why the name enzyme linked is coming. Okay. This name enzyme linked, sorry, the name enzyme linked is coming because the antibody is attached to its enzyme. Okay. So, then this enzyme also you cannot see. So, when antibody I add, actually I added the enzyme linked antibody. So, if enzyme is linked and this enzyme what we use here, this is generally horseradish peroxidase. Okay. Name is very common, there are many enzymes in the world. Okay. Not every enzyme we can use, there are certain point for that, but the enzyme we use is horseradish peroxidase. It is actually peroxidase is the enzyme and horseradish is the source, it is a plant material, it is no horse, it is a horseradish is a plant, there is no animal business, though its name is horse. Okay. Another is alkaline phosphatase. Why these two enzymes? Because these enzymes are very stable in room temperature, you do not need any special incubator for that, you can do the whole thing in just room temperature, they are very stable, they are active, and not only that, both the enzyme has a very good property because they have, this enzyme has a substrate commercially available which is colorless. Okay. Sorry. This enzyme has a substrate which is colorless, okay, but they can give a product which is colored, Okay. So, they have a substrate which is colorless, but their product is colorful. Not only that, they have a product which is water soluble. So, that means they have a water soluble colored product. That is why these two enzymes, they are very stable in room temperature, they can work, they have a substrate colorless and colorful product. So, why this is important? Because what we do is after adding that we add this substrate here and incubate in solution. What will happen? After certain time, maybe 10 minutes, 15 minutes, 20 minutes, you see the color is developed. So, what we will see actually, we will see that the well which was nothing, that solution, suppose this solution is green, okay, that solution is green, and that then whole solution will be like green here, okay, whole tube will be green, and depending on how much, I mean, so now the question is, this is the whole technique. So, you immobilize the enzyme, I mean antigen, then you add your serum, and serum contain antibody which is attached with enzyme, and then if you add the substrate, it will give color and colored solution because it is water soluble. Okay. Now, The question is, if there is no antigen, there no antibody will bind. If no antibody, no enzyme is there, no color. That means, if I am working, suppose I am checking the antigen, whether some solution has antigen or not. So, in that case antibody is known, antigen is unknown. So, one solution is given. Okay. Suppose this this water bottle. So, I am saying this water bottle has some antigen, I would like to check. Okay. So, some contamination is there. So, what I can do is I can take little bit of this water, add this in that tube, in that well, incubate for some time, and then the antigen I am looking for, I am saying that this water contains some typhoid bacteria. So, do not give me, I challenge there is typhoid bacteria. So, that means some typhoid antigen will be there. So, if I take that solution, isolate that bacterial extract and add it, if and some of you will say no, this water is completely pure, there is no bacteria, I would like to check. What I can do is I can take that water, extract the protein, add it. So, then repeat this thing, whole thing, and the antibody I will give anti-typhoid antibody. Clear? So, if this water has the antigen, it will, this red thing will come, and if this red thing comes, and then your antibody will bind, and at the end if I will see the color. If color develops, then I am right. That means, this particular water that I was showing you has the typhoid bacteria, but if there is no color developed after repeated experiment, that means there is no antigen. No antigen means there is no antigen in the water. Clear? So, this way it can tell you that whether this particular well has antigen or not. Same way, if antigen is fixed, I know antigen is pure, and definitely antibody will, you can test. So, if antigen I am sure it is there, if serum or antibody solution which is unknown, if I add it and gives the color, that means you have antibody in your solution. If there is no antibody, that means no color. So, either one we can figure it out whether antigen is there or not. Then at one thing you have to is supposed to be known. If antigen is known, antibody you can measure, is antibody is known, antigen you can measure. Now, depending on how many antigen is there, the intensity of color will change, because more antigen, more antibody, more enzyme, that means more color will develop. Okay. So, one enzyme can have multiple reaction. Same way you may have 10 molecule of antigen, or I mean let us see how many, 1, 2, 3, 4, 5, 6, 3, 3, 3, 9 antigen molecule I have added. So, now if your solution has 5 antibody molecule, so then what will happen? Then it will happen, then suppose this is not there, this is not there, this is not there, only say this 5 will be there. So, if these 5 is there, what will happen? Automatically your green part will be less green solution. So, depending on the intensity also you can tell how much antigen or how much antibody is there, because spectrophotometry we can measure this solution, measure the spectrophotometrically, measure the intensity of this solution. Everything is standard, substrate is standard, everything is commercially available. So, you do not have to do much. Okay. So, that way ELISA can tell you the amount. How you can tell the exact amount? You just have to do a standard curve. I am sure you did some DNA estimation or protein estimation in biochemistry lab. Okay. In that what you did? First you did a standard curve, known amount. So, what you have to do is you have to do basically you have to make multiple well, okay, where you have to give known amount of antigen, say 1 microgram, then 2 microgram, then 4 microgram, then 8 microgram. Okay, known amount of, suppose I am measuring the antigen. So, known amount of antigen if you give, what will happen? Every one, and then make a plot. This is OD versus this is concentration or amount of antigen. Okay. This is antigen concentration. Okay. So, in 1 microgram in this one you will get some OD, plot it. 2 microgram you will get some, 4 microgram you will get some, 8 you will get some. So, what will happen? You will get a straight line. It may go with through the origin, it may not go through the origin. Then in one solution, this one, this is say unknown, okay, that you do not know. This is the unknown that you want to measure. What you can do is, you will get some OD, say x, that x OD I am taking another color for. So, that x OD suppose come here. This is known standard, this is called standard curve. So, if unknown OD come here, what we can do is, this is the OD, and if you go back from the slope also you can calculate. If you go back, whatever the concentration you will get here, this is the concentration of antigen here. So, that is how we can very closely measure the amount of antigen present in a given solution, and most of the hormone we measure now in pathological laboratory, we use ELISA. You might have heard the HIV detection is through ELISA, the primary screening, whether it is there or not, ELISA test is so common. So, that is how it is. So, if anything is there, you will get color, if it is not there, no color. So, having color means it is there. Okay. So, this is more or less the primary part that what is there. So, in this case what we have is, in this well we had antigen. Okay, I am just now drawing one just for easy. You had antigen, then we had antibody which was labeled with a enzyme, but we can do second. This is when the antibody, the first antibody is attached with enzyme, we call it direct detection. Okay. It is called direct detection, but sometimes it is not always possible, because you isolate the antibody, the serum, labeling it with enzyme, it may or may not be possible in all the labs. So, there is no problem for that also. What you can do is, you can buy antibody, that is why I was talking about antibody, anti-idiotype in the previous class. So, you can buy, suppose this is the antibody which is raised in rabbit, okay, rabbit antibody. So, I can buy anti-rabbit, suppose this is rabbit IgG, you raise the antibody in rabbit, and after booster, first booster is mostly IgG. So, you can buy anti-rabbit say goat IgG. That means, rabbit IgG was injected in goat and antibody was raised. So, if this is the antibody, if this color is say red, no, not red, say green. So, if this color of this antibody is green, where it will bind? It will bind to this antibody, because it is raised that way. So, this antibody you can buy commercially, it is commercially available. You can buy anti-rabbit, anti-human, anti-goat, anti-mouse, whatever antibody you want, you can buy commercially. And not only the only antibody, you can buy anti-rabbit goat IgG attached with enzyme. What enzyme? Whether you would like HRP or alkaline phosphatase, up to your choice, no problem, because both are commercially available. And it mostly what happened actually, it depends which lab you are working, what is their availability. In my lab HRP is working, so if you join in my laboratory you will work with HRP, but generally it is same, but definitely there are certain advantage disadvantage of both the enzymes, but that is not very important for estimation, I mean in this kind, for this class. So, this enzyme linked secondary, this is called, so this one called primary antibody, which is my major interest. This is primary antibody, also designated like 1 degree, and this is called secondary antibody, or 2 degree. So, this secondary antibody which is enzyme linked, same. Now, just one step extra and 1 hour extra, then this is same. Now, if you give substrate, it will give product, you will get color, And this kind of using secondary antibody to do ELISA is called indirect detection. Indirect detection. First one is direct because the primary antibody was labeled with enzyme. This is second. Okay. And this is actually the simple principle of ELISA. So, the ELISA that we just discussed, this is a straightforward ELISA. Either, I mean, you can do both way. You can first immobilize the antigen, or you can immobilize the antibody, then you can do. But very common ELISA, which is called sandwich ELISA. That's what is this sandwich ELISA? You know sandwich, right? That we eat. So, what is there? 2 bread. Okay. Sandwich is 2 bread, and in between there you can put whatever you like. What is there in sandwich ELISA? With same well, same PVC coated well, but it is little bit more specific. Why it is more specific? Say suppose there is one antigen has 2 different epitope, that now you know, one is circular and another is triangular, say it looks like that. So, now if this is the kind of antigen here, what will happen first, and we are going to use 2 different antibody. One antibody can bind this and another antibody can bind this. That is the sandwich. How? So, first suppose I use this green antibody. So, I immobilize, okay, everywhere I immobilize. 3 antibody is there, just to make life simple. So, here after this 3 antibody, they are also protein, if you coat it will immobilize there, then you add the antigen. What will happen? As soon as you add antigen, okay, let me write here. As soon as you add antigen, it will bind like this. Okay. So, first you immobilize the antibody, then you add antigen, one of the epitope attached. So, if by any chance there is a mistake or some non-specific interaction, it happened because it is a protein-protein interaction, some non-specific can always happen. Just to eliminate that, to make it more specific, I am going to use another antibody. What antibody? This red one. So, now this red one bind here, right. So, this structure will happen. Okay. So, this one will happen. If you see this, this kind of structure will happen. So, then if this red which is coming second, or if I draw this flat way, so it will be better. So, one antibody here, instead of making this, one antibody here, then like this. Okay. So, then second antibody will come. I just reverse this thing. So, let me draw areas this just to avoid confusion. So, if this is the case, the second antibody will bind like this, and this is your this is the well. Okay. This is the well where everything is happening. If this particular antibody, the second antibody for example, is enzyme linked, what is going to happen? Same thing, same enzyme HRP or alkaline phosphatase, and you give the substrate and you have product here. Okay, you have product here. Principle it exactly same enzyme, same substrate, same OD you can measure, but what is this? This sandwich ELISA will give you more specificity, because you are using 2 antibody to detect one antigen. So, by any chance if one antigen one antibody non-specifically bind, another will not. Okay. So, suppose one antibody, suppose in this case here in this case suppose this antibody okay, bind something like this, but the other epitope is not triangular, but square. Okay. So, if this is the by mistake it binds, the other one will not bind, because the green one will not bind to the square. So, even in the primary time this thing happened, the second antibody is not specific to this. So, it will not bind. So, you can eliminate, but if you do the single, that all 4 will be detected, but here as we do the sandwich, one will be eliminated, because the second antibody will not recognize the antigen which is not really you want. That will increase the specificity, or it will give you more accurate result in comparison to single antibody. Here also you can do direct, indirect. This is called sandwich ELISA. Is that clear? So, here also you can have standard curve, you can have this estimation, detection, but normally sandwich ELISA we use for accurate estimation. Is clear? So, this is ELISA, but here what we can do? We can measure antigen concentration, we can detect antigen in a given solution, we can measure antibody concentration in a given we can measure antibody concentration in a given solution, or we can detect whether antibody is present in a given solution or not. Okay. So, this is actually ELISA what we call. Okay. So, this is for today. In next lecture we are going to discuss about immunoblotting. Okay. Immunoblotting is very similar principle, but slightly different. It is also known as Western blotting. Okay. So, see you in the next lecture.